And here they are at higher magnification.
MRI is an amazing piece of technology, and one that we're just getting started with. With new MRI advances, we're able to see truly fine detail from individual brains of living people. And not just at the surface, but through the whole brain. This is the hippocampus, a brain region which allows you to create new memories. Using another MRI breakthrough, we can see the wiring between brain structures, allowing us to map out entire brain-wide circuits. And while this is incredible, it's only a small piece.
This 3D reconstruction shows Purkinje cells in a living mouse that were labeled with a genetically encoded indicator to make them detectable. Here we see a side view of Purkinje cells just beneath the surface of the brain. The pear-shaped structures are the cell bodies, which connect to fan-like dendritic trees going upward and long, thin axons reaching downward. When the image rotates to show the top and bottom views, the distinctive flat structure of the dendritic tree is clear. This alignment is thought to optimize communication.
The function of the brain in its different regions, you can help to localize disease processes. We are looking at the frontal lobes of the brain. Here is the right and the left hemisphere, and between them is the interhemispheric fissure. And as I lift the brain slightly, the area of the frontal lobes is sitting on my fingers, and the frontal lobe extends all the way back to the central sulcus, which is a line that goes on either hemisphere between two gyri that tend to parallel each other. The area in front of the central sulcus is called the frontal lobe. And the area behind it, back about this far to a little notch, is called the parietal lobe. If we look at this on a lateral view, you can see the same landmarks. Here is that central sulcus separating frontal from parietal. Here is the notch separating occipital lobe from parietal lobe. And if we arbitrarily draw a line across here from this lateral fissure, we have the temporal lobe. So we have temporal lobe, occipital lobe, parietal lobe, frontal lobe. Now what I want to do is to talk about the functions of those different areas. Now I am showing you the areas and lobes of the brain with respect to their functions. So from the central sulcus on this lateral view of the left hemisphere, everything in front of this is frontal lobe, and this gyrus that parallels the central sulcus, because it's in front of, the precentral gyrus is primary motor cortex. And motor function is what you want to associate with the frontal lobe. Behind the central sulcus, the other parallel gyrus is the postcentral gyrus. And it runs parallel, and it too has a function. It is sensory. It is sensory from the body and from the face. The posterior region, the occipital lobe, you want to associate with vision from the opposite half of each eye. In other words, if this is left hemisphere, it is receiving information from the right half of each eye, or the right half of each visual field. We have here a person who is looking out at their visual world, and we're hovering, looking down from above. And we put up this blue wedge, which shows what the left eye sees when the right eye is closed. And we put up a red wedge, which shows what the right eye sees with the left eye closed. Now we both open both eyes, and we see both the red and the blue wedges. The central area between 60 degrees is seen by both eyes. This is how we get stereoscopic or binocular depth perception. However, if the left occipital pole of the brain, which represents the right or red part of your visual world, is damaged, then the black lesioned wedge between 60 degrees and 95 degrees is lost. However, with both eyes open, the patient may be totally unaware of this visual loss. That is why it is important to test each eye separately in order to pick up a massive visual field loss with an occipital lesion. Down here in the temporal lobe, we want to associate that with audition, but most importantly with interpretation and understanding of language. This area here, coming around like this superior and posterior part of the temporal gyrus is called Wernicke's area, and it is devoted to the interpretation and comprehension of language. If we understand language, we also speak language. Speech is a motor function, so it should not surprise you that an area in front of the precentral gyrus, again in motor cortex, is involved with the production of language. So we have two language areas, a temporal parietal region called Wernicke's for comprehension, and a motor expressive area in the frontal lobe called Broca's area. This specialization for language in 97% of the people is a left hemisphere function. Let me turn now and look at the right hemisphere. If the left hemisphere is specialized for language, is there something important going on in the right hemisphere? Again, our landmarks, the central sulcus with parietal lobe. The somatic sensation is right behind the central sulcus, but this area of parietal lobe, this general region of parietal lobe, is very important for body image, for your perception of your body and its relationship to space and geometry. So people with difficulties here have problems navigating, have problems recognizing or feeling the opposite side of their body, and have quite often get lost. Because language is so important and language is not in this right hemisphere, lesions or disease of the right hemisphere is oftentimes much harder to detect and only can be recognized when the disease process has progressed or the lesion has gotten very large. In comparison, lesions or damage to the left hemisphere, where the interpretation of speech or the production of speech, these areas are so critical and important to us. If these regions are damaged, it's very easy to pick this up. Back to a frontal view of our brain. If I were to take my knife or my probe and cut across, I would have, as you will see now, a frontal section through the hemisphere. This is the left hemisphere, and this is the right hemisphere. We have our cerebral cortex and gray matter, and we have our sulci and our gyri, or gray matter, going over the surface. What I want to point out to you is, let's imagine that this section came right through the motor cortex, right through that precentral gyrus. And now I want you to realize that on the surface of this, we can map a human being, so to speak. If this is motor, then from the lateral sulcus here up to the interhemispheric fissure, we have a nice orderly display of larynx and mouth and lips and hand. Very big area devoted to this in humans. And then a smaller trunk and leg and foot and genitals.
cells, or neurons, in the brain. Neurons constantly communicate with each other through electrical charges that travel down axons, causing the release of chemicals across tiny gaps to neighboring neurons. Other cells in the brain, such as astrocytes and microglia, clear away debris and help keep neurons healthy. In a person with Alzheimer's disease, the most basic form of dementia, toxic changes in the brain destroy this healthy balance. These changes may occur years, even decades, before the first signs of dementia. Researchers believe that this process involves two proteins called beta amyloid and tau, which somehow become toxic to the brain. It appears that abnormal tau accumulates, eventually forming tangles inside neurons. And beta amyloid clumps into plaques, which slowly build up between neurons. As the level of amyloid reaches a tipping point, there is a rapid spread of tau throughout the brain. But tau and beta amyloid may not be the only factors involved in Alzheimer's. Other changes that affect the brain may also play a role over time. The vascular system may fail to deliver sufficient blood and nutrients to the brain. The brain may lack the glucose needed to power its activity. Chronic inflammation sets in as microglial cells fail to clear away debris, and astrocytes react to distressed microglia. Eventually, neurons lose their ability to communicate,